In today's class, we are going to look at the use of an enzyme as an analytical tool in the hydrolysis of urea. We'll start by looking at what urea is. So first of all, a urea is a colorless crystalline compound which is the main nitrogenous breakdown product of proteins metabolism in mammals and is excreted in urine. So this is the colorless crystalline compound of the urea seen here. The next is urease. First of all, what you have to notice is that urease is an enzyme that catalyzes the hydrolysis of urea into carbon dioxide and ammonia. And ureases are found in bacteria like Lactobacillus species, Proteus species, Aspergillus, Helicobacter pylori, and Staphylococcus saprophyticus. Also in fungi, and we have yeast as an example. It can also be found in algae such as the blue algae, and also in some plants, and we have the jack beans, soya beans, watermelon, mulberry, and pumpkins as example, and also in some invertebrates like mollusks and the California sea hare, and also in soils as a soil enzyme. And one key thing that we have to notice is that urease activity increases the pH of its environment as ammonia, which is basic, is produced. Urea hydrolysis. So first of all, what we have to notice is that urea is broken down by the enzyme urease in the presence of water to ammonia and carbon dioxide. The ammonia produced in an aqueous solution will react with water to produce ammonium ions and OH ions, which causes alkalinity of the medium by increasing the pH. And this is the chemical equation showing how urea is being hydrolyzed by the enzyme urease into ammonia and carbon dioxide. Measurement of urea concentration. Urea concentration can be measured either by a direct method or an indirect method. The direct method, which is without the use of an enzymes, and the indirect method involves the use of an enzyme and basically urease over here to hydrolyze our urea into ammonia before subjecting it to a chemical method. So let's start by looking at the direct method for urea determination. What we have to note here is that the direct method for measuring urea does not involve the use of enzyme to first hydrolyze urea before quantifying its content, but rather it involves the direct chemical quantification of urea by calorimetric method as it measures the chromogen or the color produced directly from urea. And examples of the direct method for urea determinations include the Kejel method, the diacetyl monosime method, the Ferrins method, and the autotaldehyde method. The indirect or the enzymatic method for urea determination. The indirect or the enzymatic method for measuring urea involve the quantification of the ammonia produced through a chemical method after urea is being hydrolyzed by the enzyme urease. And the chemical reactions or methods used here are the Scott method, the Bertlet method, the Nestle's method, and the glutamate dehydrogenase method. So these are all examples of methods that are normally used in the indirect or the enzymatic determination of urea. And also, the ammonia produced in the enzymatic method can be quantified in two ways. One, the endpoint method, which makes available conditions in titration to quantify the amount of urea completely decomposed into ammonia by the enzyme urease. And two, a reaction method or a reaction rate or a kinetic method where the product formation is monitored over a fixed time to determine the rate. The great advantage of the enzymatic method of identifying and estimating substances is that enzymes are more specific than the direct or the usual chemical method. Example, the total nitrogen in urine can be estimated by the Kegel method, which is a direct method, but these will measure nitrogen in ammonia, amino acid, proteins, uric acids, as well as urea. By the use of the Scott's method, which first involved the use of the enzyme urease to break down urea into ammonia and then titrated is specific and helps to quantify the exact amount of urea. Let's look at some factors affecting urease activity. So the first has to do with the pH, the second temperature, the third substrate concentration, and fourth inhibitors. And over here we have let us look at some example. So we have the 2-beta mercaptoethanol as a competitive inhibitor. We also have the fluoride as a mixed inhibitor. We have the acetyl hydrozamic acid as a competitive inhibitor. We also have the bicalin as a competitive inhibitor and iminothioxylene sulfonamide as an uncompetitive inhibitor. And these are the factors that normally affect urease activity. Now, let's look at the Scott's method. So first of all, what you have to note is that the Scott's method is specific for urea analysis because the enzyme urease is used in this method to break down urea to produce ammonia only, which can be titrated and quantified. Principle of the Scott's method. Urease activity increases the pH of a medium if ammonia is produced, and this makes the solution basic. For that reason, the pH is adjusted using a specific mixture of indicators. A mixture of urease and urine is then incubated to hydrolyze urea to liberate ammonium carbonate, which is titrated against the acid HCl. And as I explained earlier, so the advantage of the enzyme method of identifying and estimating substance is that enzyme is much more specific than the usual chemical reagent. An example, the total nitrogen in urine can be estimated by the Kegel method, but this will measure the nitrogen in ammonia, amino acid, proteins, uric acid, and etc., as well as the urea. So you end up not being able to quantify the exact amount of the urea present. So now let us look at some indicators that are normally used in the Scott methods to quantify the amount of urea present in a substance. So the first is the bromocresol green indicator. So first of all, what we have to note is that the bromocresol green is a triphenyl methane dye that is used as a pH indicator for titration. and also used as a pH indicator in DNA, agarose gel electrophoresis, and microbiological growth media. So its chemical formula is this, and the bromocresol green is a pH indicator that is yellow below pH 3.8 and blue above pH 5.4. So as you can see over here, so any pH at pH below 3.8, it turns yellow, and at pH above 5.4, we can see that it turns blue. And in between the pH 3.8 and 5.4, it is green. So as we can see over here from the pH color chart, we can see, so between the pH 4.0 and 5.0, it has a different shades of green as seen here. The indicator is made from bromocresol green powder dissolved in ethanol. The next indicator is the methyl red. So what you also have to note about the methyl red is that the methyl red is an azo dye or a synthetic dye, which is one of the common chemical indicators used in the laboratory to determine pH transitions within a specific range. It's most effective between the pH of 4.4 and 6.2. And if the pH level is below 4.4, the color turns red as seen over here. And if it is about 6.2, the color turns yellow. And this is the pH range at which the methyl red indicator effectively works as seen here. And this is the molecular structure for the methyl red indicator. So now let us look at the test protocols for our experiment. So we'll start by looking at the materials or the reagents needed for this test. The first is 0.03 molar HCl. We also have the indicator, and the indicator comprised of 3 parts of 0.04% methyl orange and 1 part of 0.4% bromocresol green. And also urine sample, which is to be donated by student, and burette and stand and water bath set between 40 to 50 degrees Celsius. And some important things that we have to note here is that urease activity increases the pH of its environment as ammonium is produced. And 2, methyl red is a sensitive indicator for titration of weak organic base and ammonia, and it is an acid-base indicator. Also, the bromocresol green indicator is an acid-base indicator which is used as a tracking dye, and it exhibits a color change within the pH range of 3.8 to 5.4. And HCl is useful agent because it forms amide bonds in an aqueous medium. Now, so let us look at the test protocol or method. So first is to set up 6 test tubes. So this is the first test tube, second, third, fourth, fifth, and sixth. So the first test tube should contain 2 ml of our urease enzyme plus 5 to 6 drops of our indicator. The test tube 2 also contains 2 ml of urease enzyme plus 5 to 6 drops of indicator. The third test tube contains 2 ml of our urease enzyme plus 5 to 6 drops of indicator. The test tube 4 contains 2 ml of our urine, which has 1 to 10 dilution to 5 to 6 drops of the indicator. The fifth test tube contains 2 mils of the diluted urine and 5 to 6 drops of the indicator, and the sixth the sixth test tube that happens to be our control contains 2 mils of distilled water plus 5 to 6 drops of indicator. And one thing that we have to note here is that color tends to greenish blue in each test tube after the addition of the indicator to each of the test tubes. So 2. The second step is to add to each test tube drops of the HCl until the first tinge of red appears. And what we have to note is that the appearance of the first tinge of red changes the solution in each test tube from greenish blue to mauve color. So this color is an indication of the appearance of the first tinge of red. So thus, the formation of a mauve color indicates the presence of the first tinge of red. And if too much HCl is added, the enzyme urease is used to adjust the medium. Step 3. So we mix solution. So that's we mix test tube 1 and test tube 6. So we pour we add the components in test tube 6 to that of test tube 1, and also for test sample 1 and also for the test sample 1. So this is our control. So for the control over here, we add test tube 1 and test tube 6 together. For our test sample 1, we add test tube 2 and test tube 5 together. And for our test sample 2, we add the components in test tube 3 and test tube 4 together. So in all, we are going to have 3 different setups, with the first being the control, and this is our test tube 1 and our test sample 1 and test sample 2. That happens to be a duplicate of our test sample 1. So the fourth step, you cover the 3 test tubes with aluminum foil and incubate at 50 degrees Celsius for 5 minutes. So that's the control test tubes and 2 of the test samples. And 5, the fifth step, you allow to cool and titrate against HCl in burette. And what you'll have to note is that the endpoint color gives a brick red color as seen over here. So this is my assumed table of results after the Scott's method. So we can see so this is our final volume, this is our initial volume, and this is our title volume. So we are going to titrate each of the test sample in each test tube and compare the color change against that of the control. So as you can see from the assumed table of result over here, we have a tighter volume of 3 centimeters cube for test tube 1, and also for test tube 2 we also have a tighter volume of 3.0. So we have an average tighter volume of 3.0 centimeters. So let's just try some questions over here and see how to solve them. So the question says you should calculate the urea content in the original urine sample in 1 in grams per 100 mils and in 2 in grams of the urea nitrogen in 100 mils. So let's see how to do this calculation. So first, we will have to write our balanced chemical equation. So as we can see, we have our equation 1 and equation 2 over here. So the equation one happens to be the hydrolysis of urea in the presence of water by the enzyme to produce carbon dioxide and ammonia over here. Also, the equation 2 happens to be the titration of the ammonia produced in the equation 1 with HCl to produce ammonium chloride. So next, what we have to note is that, so let us try and compare the moles uh, of each of the reactants and the product in each of the equations. So from equation 1, we can see that the mole of the ammonia and the mole of the urea to the mole of the ammonia produces 1:2. And also from equation 2, we can see that the mole of the ammonia against the mole of the HCl is 1:1. So we will first work around the equation 2. So let's see how to do that. So that's from equation 2. We know that the moles of the ammonia is the same as the mole of the HCl. But we know that the mole of the HCl is giving us the concentration of the HCl used times the volume of the HCl obtained in the titration. So hence, this implies that so the mole of the HCl is giving us the concentration of the HCl used times the volume of the HCl in our titration. And this is 3 times 10 to the negative 3 over that. So if you are to solve this equation, we will get the mole of the HCl to be 1 times 10 exponent negative 4 exponent negative 4 moles. And also, we also have to know that since the mole of the HCl is equal to the mole of the ammonia, then what it means is that the mole of the ammonia is also the same as that of the HCl. So hence the mole of the ammonia is also given as 1 times 10 exponent negative 4 moles. And what we also have to note is that from equation 1, we also saw that the mole of the urea to the mole of the ammonia is 1:2. So this implies that the mole of the urea here is equal to the mole of the ammonia divided by 2. So hence, if you are to do this calculation, we can see that the mole of the urea here is giving us 5 times 10 to the negative 5 mole. But what we also have to note is that the urine containing the urea that we use was diluted. So we did 1:10 dilution. So hence, to determine the actual moles of the urea present, we multiply the mole obtained over here times 10. So hence, if you are to do this, we will see that the mole of the urea in our urine is 5 times 10 to the negative 4 moles. So now let us look at the molecular weight for the urea. So the molecular weight for the urea is given as 60.06 grams per mole. So to calculate the concentration of the urea present, we know that concentration is also given as mole divided by the volume. So we substitute the moles that we had over here, which is 5 times 10 to the negative 4 mole divided by the volume of the urine use. The urine use, which is 2 mils. So if you have to substitute this into the equation, you'll get the concentration of our urea to be 0.25 molar. What you also have to notice is that the volume 2 times 10 exponent negative 3 dm cube is used because 2 mils of the urine was used for the experiment. So now, let us now, from our previous calculations or from the mole ratio, let us now let us now do the calculation of urea content in the urea sample. So we'll start by looking at the gram per 100 mils. So what we have to note is that grams per mil is the same as the mass concentration. And mass concentration is given as the molar concentration times the molecular weight. So the mass concentration is given at the 0.25 times the molecular weight of the urea. So if you are to do our calculation, we can see that the mass concentration of the urea is given as 15.02 grams per dm cube. And what you have to note is that mass concentration is expressed in dm cube, or let me say 1 liter or 1,000 mils. So what we'll do is that, so if the 15.02 grams is in 1,000 mils, how many grams of the urea will be in the 100 mils? So if you are to make as a target from here, or if you are to cross multiply, you'll see that the grams of urea that will be in 100 mils will give us 1.50 gram. So this implies that 1.50 grams of urea content is in our 100 mils. Now, so the next question says we should also calculate in grams of the urea nitrogen in the 100 mils. So what it means is that the amount of nitrogen present in the urea per 100 mils. So to do this, we will first have to determine the molecular weight of our urea. So the molecular weight of a urea, so this is carbon that with a molecular weight of 12.01 plus our 16.0, which is for the oxygen, into bracket the molecular weight, the mass or the atomic number for the nitrogen, which is 14.01 plus 2 of the hydrogen. So if you are to do this, we will see that the molecular weight for the urea is given as 60.06 grams per mole. So now, the molecular weight also for the amount of nitrogen present in the urea is given as 2 times 2 times 14.01 over here. So this is equal to 28.02 grams per mole. So what we have to note is that if 60.06 grams per mole is in 1.50 grams, then 28.02 grams per mole will be in what grams? So if you are to cross multiply, we will see that, so we will get 0.7 grams in 100 mils. What it means is that the mass of the nitrogen present in the urea in 100 mils is 0.70 grams. Thank you.